Hello, everybody. Welcome to Tuesday, January 12th, 2021. It's a great day to be a junior potter. Mr. Parrott here with your rundown of what you're going to be doing on Tuesday. First off, there's a new bell ringer up there, so make sure you get that thing answered up. We're going to be finishing our final portion of our DNA RNA notes today. So we're going to be looking at number 5 on there. Which will be learning how to make a protein out of RNA. And there's going to be an assignment that goes along with that that comes later on. So, without further ado, let's look at number 5 as far as what is going on with our RNA to protein, which is known as translation. First off, our process known as translation today is going to be taking the message, delivering it to the ribosome so that the ribosome can go on to make a protein. It is going to use all 3 versions of our RNA today. You can see that down below here, this blue strand, this would be the messenger. The green little blobs that are coming in and out are going to be, or that are there that are just sliding along, that is the ribosome. And then these little pitchfork looking things, which will sometimes look like a T, will be our tRNAs delivering our amino acids to it. So let's take a look at how this process works. First off, with our message, we actually have a secret message within the secret message itself. And we first need to talk about these things called codons. A codon is short for the word or is used in place of the word code. And what it is that it is three mRNA bases that are in a row. And basically what the ribosome does is that it is going to chunk out the message into sets of 3. And for every 3 bases, there's a protein that gets brought in depending on what those 3 bases are. We call that the codon. It's like a secret message, a secret code going on there. And we have what is called a codon chart that we'll then use to see how the codon works. This would be our codon chart. And it works very similarly. Whatever your first base is, you look at what it is on the left side. So let's say we had the... Oh, I can't write on this, can I? We'll look at it in just a little bit. The second base you go off of the top. So whatever the second letter is in your sequence, you go off the top. And whatever your third base is, you go over here on the side. Let's do this. Let's see if I can make a little sticky note and drag it over here. Oh, yeah, look at that. I can make a little sticky note. Let's say we have the formation of AUG. Can I make that any bigger? I don't think I can make that any bigger. I can bold it. Well, let's say we have an AUG. That is our codon. That is our 3 letters that will make up a protein. So to show how this works, the first thing you need to do is look at the first letter. Let me pop that back up there. I clicked accidentally. So our first letter is an A. You go over here to where it says first base, an A. That means we're going to be looking for the protein that is going to be in this row. We look at what the second base is going to be. The second base is a U. So you go up to the top where it says second base. Here is U. You go down this row. And where the first and second base overlap, this is what we're going to be looking at here in this row here, or in this little box now. So we're basically going from big to small to find out where the stuff is. And where all 3 of the letters overlap, that is going to be our protein. So let's look at our third letter. Our third letter is a G. Notice that there are 4 G's over on the third base side. You'll need to find the G that matches up with the first. So since we had an A, we're going to find this G here. We bring this G all the way over to where the A and the U met up. And we have this amino acid called MET, or MET. So that would be the first part in our protein if we were creating something there. Now, all of these are abbreviations. I'm giving you the abbreviation list. They all stand for bigger and more complex things, like MET stands for methionine. You got phenylalanine in there. You got tryptophan in there. You got glutamate. You got glutamine. There's a lot of different long, complicated words that are the amino acids. We'll use them as the abbreviation. So you'll always see them in these abbreviated forms or something close to these abbreviated forms. So how our proteins are made is that we take the amino acids, we link them together. When they are linked together, they make a protein. I'm going to get rid of this part here. Bye-bye sticky note. So the actual steps that go into translation, because there's a process by which this goes in. The first step is that the message attaches itself to the ribosome, and the ribosome finds what we call the start codon, which the start codon is always the same. It is the three letters AUG. Whenever you see those three next to each other, that gives the ribosome the indication to start here when it's in those three chunks. If that chunk never arrives, the protein never gets made. Because what actually our DNA does when it makes the message, it puts a little bit of nonsense basically before it. That way the message doesn't get messed up on the end. So it reads it for a little bit until it gets to the start, and then it'll actually start the process. So the messenger attaches to the ribosome, and the ribosome then goes to find the start codon. And it's kind of nice that we have these start codon, because we're also going to have stop codons in just a little bit. Step 2 is that a tRNA brings in the correct amino acid, which remember is the protein part, to the ribosome. And they're going to match up based off of those three letters. We do the base pairing rule, so they lock it in to make sure that it is confirmed. Once they have confirmed it, they drop off the amino acid. If it doesn't match, it gets the boot, and another one comes in to see if it's the match. You might hear the word anticodon thrown around. This would be the time where there would be an anticodon happening. Step 3 is that this process repeats. So a new tRNA molecule comes in, it reads the next codon and brings in the new amino acid. So it's just going to keep on repeating itself over and over and over again. So where we get step 4, the new amino acid connects to the last one, and this process is just going to repeat. So we grow a chain of it here. And you can kind of see that chain with the red circles here. These are the protein parts. The new one comes in, boots the old one out. They connect up in the middle here to continuously grow the protein. This part will happen over and over and over again. New one comes in, connects up, leaves. New one comes in, connects up, leaves. The new one comes in, connects up, leaves. It keeps doing this process until it gets to step 5, where it continues until it reaches a stop codon or the end of the message. Either one of them could stop it. So if it runs out of the message, it can't continue on any longer, and it just makes whatever protein got made there. Or it'll reach a stop signal before the end of the sequence. So the stop codon, there's 3 of them, and on our chart, let me go back to the chart here, they literally say stop. You can see them up here in the corner, because they all start with U's. So we got 3 stops here. This is not actually an amino acid. It is literally nothing. So, or like a water droplet, or it's something that creates space so that when the tRNA comes in to try to combine up, like in this picture, there's not going to be anything here. There's not going to be an amino acid. So when the next one comes in and tries to combine up with the chain, it's going to combine up to nothing, signaling to the ribosome that it is done, so the protein then gets released to go do whatever the protein needs to do, whatever trait it needs to combine up with. And there's 3 of them, like I said. The three, you don't need to memorize these because you'll always have a chart, as UGA, UAG, and UAA, but there's three of them. They don't count for an amino acid, so they're not a part of our 20 that we have. So we have 20 amino acids. They're just simply a stop. It's a stop codon. Here's a video kind of go throughing all those different phases for you to look at and watch. Can't watch it here because that'll be a YouTube inside of a YouTube, which you're not supposed to do. Let's take a look at your assignment though. So go ahead, go to the Schoology folder, open up Tuesday. I'm going to go ahead and do that as well here. And there will be an assignment on there that says Assignment Translation Worksheet. Let's take a look at what you're going to do today. Just like all the worksheets, you got multiple attempts on it. So you'll start your new attempt. For a lot of these, I'm going to be giving you the chart, and there's just going to be questions for you to answer. Some of them being the multi-choice variety that comes from the chart that is there. And they're all randomly ordered. And then for some of them, I give you the chance to use the chart. So for this one particularly, I give you the three bases that make up a codon. You will then have to use the table up top to decode it and find the correct answer to it. So you'll have to choose out of the box which one is the correct one. Like I said, there's just some multiple choice to just understand how things work. And then the last one here, this is an entire message. How you're going to work this is that you're going to chunk it out into threes. So like your first chunk there is AUG. You find what AUG is, you put it in the first box. I'm just going to pick one. I'm not going to give you the answers here. Second grouping of three, you're going to look at. Notice how I did not include any of the first three. It is a complete, once you use a base, you do not go back to it. So the second one is GUG. So you look at what GUG is, pick up, what that is, and then go on to your next three. You then go on to your next three. For some of you, it might be easier to write this out on a piece of paper and then put like little slashes in between them. If you're able to remember to put the space in there just visually, you could do it that way. But that would be the kind of ultimate end goal of today, is that you can get a message and then be able to give me the entire protein sequence that comes to it. And this one we treat as a correct one, so it should start off with the start codon and it should end with a stop codon for that. So hopefully you can double check yourself in that kind of matter. Like always, if you have any questions whatsoever, do not hesitate to drop me a line and ask via email. If you want to get a Google Meet set up, we can set up a Google Meet. Just ask for one. We can kind of work things out that way. But I hope to see you back in class sooner rather than later. So I hope you're taking care of yourselves out there, and I will see you later.